studying brain vasculature, even starting as an undergraduate. He completed his PhD in physiology and pharmacology at Oregon Health and Science University, studying mechanisms governing the release of neuropeptides from perivascular trigeminal afferents at the brain's surface. As a postdoc in the lab of Micah Nedergaard, he led work characterizing the perivascular glymphatic system and its role in clearing waste from the brain during sleep. Since starting his own lab in 2013, Dr. Iliff's work has focused on defining the glial and vascular changes in the aging and post-traumatic brain that underlie impairment of glymphatic function and the vulnerability to the development of conditions like Alzheimer's and Parkinson's disease and chronic traumatic encephalopathy. So Dr. Iliff is a professor in the departments of psychiatry and behavioral sciences and in neurology at the University of Washington School of Medicine, where he is also the Arthur J. and Marcella McCaffrey Professor in Alzheimer's disease. He's the associate director for research at the VISN 20 Northwest Mental Illness Research, Education, and Clinical Center at the VA Puget Sound Healthcare System. He serves as lead for the UW Alzheimer's Disease Research Center Research Education component. So I welcome Dr. Iliff to our series, and we're really looking forward to your talk today. After the talk, we'll hold a Q&A session for about 10 minutes. So please, everyone, feel free to post your questions in the Ask a Question tab, and I'll moderate at the end. We'd also like to invite those who are interested in our Meet the Speaker session following the seminar for a more in-depth discussion with Dr. Iliff to please join us at the Zoom link that I'll share in the chat momentarily. So I'll now give the stage to Dr. Iliff, and yes.
All right, thank you very much for that nice introduction. Can you hear me okay?
Yep.
Okay.
All looks good.
Can you see my screen?
Yes, slides are good.
All right, we're winning already. Well, thank you for the opportunity to present to this group. I'm actually looking forward to having the opportunity to speak to a pretty diverse group of folks who maybe know something about what has been emerging in the glymphatic field over the last 10 years or so, or maybe folks who are relatively new to the space. So my goal over the next 45 minutes or so is to provide sort of a postcard from where we are at the frontier of understanding how glymphatic biology works and how its function seems to fail in the setting of age-related and post-traumatic neurodegeneration, and specifically how we are coming to understand the potential role for that biology in the development of human disease. So just to give you a sense of the biological focus of our group, we are interested generally in neurodegenerative diseases, so like Alzheimer's disease or Parkinson's or ALS or chronic traumatic encephalopathy, but we're specifically interested in the common features among neurodegenerative diseases that link them together and what the biology underpinning those common features might be. So one of the key features of neurodegenerative diseases is the presence of observable on histopathology, but now increasingly observable in vivo with techniques like with positron emission tomography or the measurement of CSF biomarkers, is the presence of misaggregated proteins. So like in Alzheimer's disease, you have extracellular plaques made up of amyloid beta and intracellular aggregates of post-translationally modified tau that form neurofibrillary tangles. In Parkinson's disease and Lewy body dementia, you see intracellular Lewy body aggregates. In ALS, you see intracellular aggregates of TDP43 and other proteins. So our basic biological question is, what is it about the brain that makes it vulnerable to this process of misaggregation? And as we think about that question, we gravitate toward non-genetic risk factors that appear to drive these diseases sort of across the board. So we think about aging. So aging is, of course, the strongest risk factor for the development of all neurodegenerative diseases, so much so that you almost don't even think about it as a risk factor. We also think about the presence of cerebrovascular disease, prior exposure to traumatic brain injury, and increasingly, sleep disruption, particularly in midlife, has emerged as a non-genetic risk factor for the development of all-cause dementia and of Alzheimer's disease in particular over just the last couple of years. So our question is, or the biological framework that we're coming to, or I should say the translational framework is, What is it in the aging brain? What is it in the injured brain? What is it in the sleepless brain that makes them vulnerable to protein misaggregation and the clinical progression, development and progression of diseases like Alzheimer's or Parkinson's? The biology that we focus on is the biology of brain interstitial protein homeostasis, which in reality is the actual topic of this talk. Now, it's not the title that I gave because I think probably 0 people would have come to this talk if this was the title that I listed. But really, everything that we're talking about is about interstitial protein homeostasis in the CNS, in brain tissue. Now, if you learned anything about how we think about how proteins in the brain's extracellular compartment are managed and maintained, you probably learned some version of what is termed the CSF sink hypothesis, which was articulated by Hugh Davison in the middle of the 20th century. And this framework was based on an anatomical observation that the CNS lacks lymphatic vessels, right? And so the lymphatic functions, or the functions that lymphatic vessels subserve elsewhere in the body, such as the reabsorptions of interstitial fluid and protein, the process of immune surveillance, those lymphatic functions have to be subserved in the CNS through some other process, through some other process that doesn't involve lymphatic vessels. And so what was proposed in the middle of the 20th century was that the CSF, the cerebral spinal fluid, serves this quasi-lymphatic function. So the CSF is a relatively protein and cell-free, clean, clear fluid that's secreted by the choroid plexuses of the lateral, third, and the fourth ventricles. It flows out through pulsatile bulk flow through the ventricular system and spills out into the subarachnoid space that surrounds the brain. So the idea is, and that CSF is then at least classically thought to be reabsorbed into the venous drainage across arachnoid granulations or arachnoid villi, something that sort of a classical understanding that has come into revision in just the last couple of years. And so the idea was that proteins and solutes within the brain tissue, between the brain cells, that can't be locally degraded and can't be effluxed across the blood-brain barrier, the way that those are managed is through exchange, either with internal CSF compartments of the ventricles or with the external CSF compartments of the subarachnoid space. And through that process, which was thought to be relatively diffuse, so disorganized and slow, that's the way that the brain maintained interstitial protein homeostasis. So now this group in particular probably is aware that pretty substantial elements of this framework have come into revision in just the last couple of years. So two key elements of this have undergone a lot of rapid development. So the second chronologically, but the first that I'll talk about real briefly here, is this notion that the CNS lacks lymphatic vessels. So beginning in 2015 in Joni Kipnis's lab and in Kari Alitalo's lab in Helsinki, there occurred first in mice and more recently in human and in the primate brain, the characterization of what to all appearances are true lymphatic vessels. Now, it still remains true that these lymphatic vessels do not appear to be within the CNS tissue itself, right? So there doesn't appear to be evidence for lymphatic vessels within the brain or within the spinal cords. But what is observed is that there do appear to be bona fide lymphatic vessels embedded within the dura, so one of the meningeal layers that surrounds the brain, either associated with the dural sinus structure, so the large veins at the surface of the brain, or down within the skull base and associated with cranial nerves. These are functional lymphatic vessels, so solutes that are injected into brain tissue or are present in the CSF are taken up into these lymphatic vessels, and it's widely believed that this meningeal lymphatic uptake supports the clearance of solutes out of brain tissue and out of the CSF, down the cervical lymphatic drainage, and eventually out into the blood. This is a very interesting biology. I know that this group has probably heard something about it. I'm actually not going to really talk much more about it. What I will be talking about for the duration of for the remainder of this talk is this notion that that the exchange between the CSF that surrounds the brain and the interstitial fluid between the cells of the brain, that is a slow and diffuse process and a non-regulated process rather than a rapid, organized, and physiological process. And this is the biology of what has been termed the glymphatic system, which was characterized beginning in 2012 and 13. Um, this is there's a little bit of hazard in talking about both of these things, in that they were described separately, right? So for the glymphatic biology was articulated between 2012 and 14. The meningeal lymphatic vessels were initially characterized in 2015. The literature as it emerged, just sort of historically, suggests that these are separate processes, but I don't think that most of the folks operating in the field now think that that's the case. In fact, I think that a reasonable way to think about this is that these are two linked parts of a general process for the clearance of solutes out of brain tissue, a process that involves essentially two exchange steps. So one exchange step by which solutes from the brain interstitial compartment move out into the CSF, that along perivascular spaces or through the process of perivascular exchange, that's glymphatic exchange. And the second whereby those solutes in the CSF are taken up into meningeal lymphatic vessels and drained through the cervical lymphatic drainage. So I don't really, even though they were described separately, I don't think that that these processes should be necessarily regarded as separate. So how do we know then that the exchange between the CSF and the brain interstitial compartment is anatomically organized and rapid? How have we come to understand this? And the straightforward answer is that we know this because we can now see it. So that I think the big innovation that has allowed this field to move forward from where it was in the 80s and the 90s was the emergence of imaging techniques that allow these fluid exchange processes to be visualized in vivo and dynamically. So this is a very straightforward experiment that gets at the basic anatomical finding that we have, and that is if you inject a fluorescent tracer. In this case, it's bovine serum albumin, so it's about a 65 kilodalton tracer into the CSF in the subarachnoid space, so into the cisterna magna, which is back behind the cerebellum on top of the pons. If you inject it in, wait about 30 minutes, fix the mouse, pull out the brain, and look to see where the CSF tracer has distributed, you can get a sense of how CSF and the solutes within it are moving over the brain surface. And what you see immediately, just by looking at the images of the injected mouse, is that the CSF isn't moving through over the brain surface and through the subarachnoid space diffusely or uniformly, but is rather following very specific anatomical pathways. So you can see that it moves initially along the ventral surface of the brain, along the communicating arteries of the circle of Willis. You can see that it follows outward along the vessels originating at the circle of Willis, like the middle cerebral artery that moves up and over the convexity here. You can see, if you zoom in, you can see that the subarachnoid CSF tracer moves out along these arterial segments all the way out to these little dots, which are the places where the surface arterial vasculature makes a 90-degree turn and dives down into and through the cortical mantle. What you can appreciate just by looking at this single image is the importance that the arterial vasculature serves in scaffolding the movement of CSF over the brain surface. So you can see here the course of the middle cerebral artery, and you can see the corresponding caudal rhinal vein, which is draining that same region of the cortex. And you can see that the organization of fluid and solute movement over the brain surface is organized along the arterial but not the venous vasculature. So that's an organization that is set up at the level of the leptomeningeal vessels that surround the brain, but then persists through brain tissue. So we can process that, or we can follow that process down into brain tissue using in vivo 2-photon microscopy. So this is a similar experiment. The key difference here is we're imaging the movement of tracer into the brain of a living animal. We're imaging, in this case, through a cranial window in an animal that's been injected with a 40 kilodalton fluorescent green dextran that's been injected into the CSF, and then the blood vasculature has been counterlabeled with a red dextran. And what you can see here, this is essentially the same thing I just showed you, which is that the CSF tracer moves along the cortical surface, along the outsides of the arterial vasculature. So this is a Peel arterial here that crosses over a Peel vein, and you can see that the tracer arrives in our field of view initially along this arterial segment, and then it gains access to the wider vasculature from that arterial segment. If we turn our view down into brain tissue, what you observe is that the same process that starts at the brain surface doesn't end there, but rather continues down into and through brain tissue. So you can follow the course of this penetrating vessel as it moves from the brain surface down through the cortical mantle here. And what you can observe is this little half moon of green, which is what's termed a perivascular space, or a Virchow-Robin space, which is actually a fluid-filled space that surrounds a penetrating vessel. And you can see that the CSF tracer is following down along that penetrating vessel, and it begins to exchange through the surrounding interstitium. So this green haze that you see here is the CSF tracer that started back behind the cerebellum, made its way over the ventral surface of the brain, followed the middle cerebral artery over the convexity, and has now begun to move back into and through brain tissue, and is now sort of beginning to fill the brain extracellular compartment. So this is a microscopic view of perivascular exchange that's happening at the level of single vessels, but we see that the same process is unfolding across the entirety of the brain structure. So this is a different imaging modality, but it's essentially the same experiment, right? So here we're imaging a rat, but not a mouse, rather than a mouse. We're using MRI instead of fluorescence imaging, and we're injecting an MRI contrast agent, gatoteridol, instead of a fluorescent tracer. But everything else is basically the same. So what I've done is I've encoded the signal change resulting from the presence of the contrast agent in blue that occurs over the first hour, the signal change that occurs over the second hour in green, and the signal change that occurs over the third hour in red. So you can get a sense of a time course from a single image. So after injection of the gadolinium into the CSF at the cisterna magna, you can see the earliest movement along the ventral surface of the brain. You can see the initial movement in this first hour I should say in the second hour in green into brain tissue is occurring along the courses of large resistance arteries, like the anterior cerebral artery, which is visible here on this coronal slice, the choroidal arteries, which you see here in this coronal cut, and the posterior cerebral arteries, which you can see here in this axial view. And then in red, you can see the more slow and dispersed movement of contrast agent through brain tissue. A movement which is organized, whose inflow is organized around these large arterial segments, but then proceeds diffusely and slowly through brain tissue toward the deep venous drainage of the great vein of Galen and the straight sinus and the confluence of the sinuses. So there's this arterial organization to the movement of CSF and solutes into and through brain tissue that is organized around the cerebral vasculature. All of those things that I just showed you, these are in rodents. And of course, while rodent biology is very interesting and informative, it matters whether this biology holds true and is present in the human brain as well. So a group in Oslo, so Geert Ringstad and Per Christian Eide, a neuroradiologist and a neurosurgeon there, have conducted, beginning in 2015 and continuing to the present day, a series of seminal studies in human subjects doing essentially this experiment we did in rodents, but in people. So what they do is they inject gatobutrol, so a different gadolinium-based contrast agent, intrathecally, so into the CSF at the lumbar spinal cord. They wait and image these subjects dynamically over time. So after the first hour, you can see that the contrast agent fills the CSF spaces, so the cisterns at the base of the skull. You can see early movement of contrast frontally, but you can start to begin to see its penetration into brain tissue. And when subjects are brought back the next day for imaging, what you see is CNS-wide enhancement that represents the movement of this CSF contrast agent into and through brain tissue, much the same way that we see in rodents, in mice, and in rats, suggesting that this perivascular exchange between the CSF compartment and the brain interstitial compartment isn't restricted strictly to rodents, but is also happening in the human brain as well. Okay, so a question that has arisen along the way is whether this glymphatic function, whether this perivascular exchange between the CSF and the brain interstitial compartments, whether that's constant across different physiological states. And this is a paper that I think has been well enough covered, so I'm just going to review it very quickly. We evaluated whether glymphatic function was constant between waking and anesthetized, or waking and sleeping states. So this is a very similar experiment to what I showed you before. So here we're imaging down into the cortical mantle. So this is an image taken from inside brain tissue. The blood vasculature is counter-labeled in this case in blue. The only difference between this this video here and what I showed you before is this animal is actually awake while this imaging is taking place. And it looks like the video isn't playing, but it actually is. It's just that there's very little CSF tracer movement into the brain while the animal's awake. But if you take this same animal and then you anesthetize it, and then you continue imaging, so this is the same animal just a short period of time later, you see that the situation in the anesthetized animal is fundamentally different than what you see in the waking animal. So the brain has shifted from a situation in the waking state where exchange is relatively slow to a situation in the anesthetized state where the exchange is much more rapid. And you can see that plotted here when you compare anesthetized animals to awake animals with much more rapid CSF tracer influx into animals that are anesthetized compared to waking. Now, an anesthesia or sedation is not the same as sleep. So in a parallel set of studies, we evaluated the exchange between the CSF and interstitial compartments in animals that were sleeping on the 2-photon scopes and animals that were waking. We saw the same sort of thing, with more rapid exchange occurring in animals that were asleep compared to those that were awake. This isn't simply restricted to the movement of CSF tracer into brain tissue, but it also extends to the clearance of solutes out of brain tissue. So in these experiments, and in the experiments I'm going to talk about over the next 20 minutes or so, what we do is we inject a radio-labeled tracer, whether an inert tracer like inulin, which is a molecule, it's about 5 kilodaltons, it's a polysaccharide. It doesn't cross the blood-brain barrier, and it isn't taken up by cells. So its only way out of the brain is through clearance to the CSF compartment, or we can inject a more biologically relevant molecule like amyloid beta. So in this case, we inject radioiodinated amyloid beta 40 into the brain, and then we measure its clearance out of the brain over time. So if we look at the clearance of inulin between waking, sleeping, and anesthetized animals, we observe that the clearance in the sleeping and the anesthetized state compared to the waking state is much more rapid. And if we convert that into a rate constant, you can see that the rate of clearance from the sleeping and the anesthetized state is more than twice what we see in the waking state. If we measure the clearance of radiolabeled amyloid beta, we see something similar, with about twice the rate of clearance in sleeping or anesthetized animals compared to waking. So what these data tell us is that the exchange between the CSF and the interstitial space, so both the movement of CSF into brain tissue and the clearance of solutes out of brain tissue, appears to be regulated by the sleep-wake cycle, and that certain anesthetics, in this case, this is a cocktail of ketamine and xylazine, can recapitulate the sleep-active clearance that we observe. So paralleling what I mentioned before, if this is something that's happening only in the rodent brain, while interesting, it's of limited interest. And so actually this paper that came out in Brain, it was probably about 3 or 4 months ago now, from the group in Oslo, was very important in that it was the first in vivo demonstration in humans that this sort of sleep active clearance of solutes out of the brain was actually occurring, not just in rodents, but in the human brain as well. So again, the group here is injecting gadobuterol, so a gadolinium-based contrast agent, intrathecally into the lumbar interthecal space, so the CSF space around the lumbar spinal cord. Then the subjects are imaged every couple of hours over time. So they took 2 groups of people and did these injections, and then imaged them 5 to 7 hours later. And they saw a relatively equal loading of contrast into the brains of people between the 2 groups. One of the 2 groups was then allowed to sleep normally overnight, and the other group was kept up all night. It was enforced sleep deprivation overnight. And then they were brought back the next day, and the clearance of solute out of brain tissue was evaluated. And what was observed is that the subjects that were permitted to sleep normally overnight, the clearance of the contrast agent out of brain tissue was more rapid than in the subjects that were kept up all night. And interestingly, when the subjects were then allowed to go home and sleep normally on the second night, the subjects that underwent sleep deprivation still hadn't caught up in terms of contrast clearance out of brain tissue, suggesting that it takes more than 1 night of normal sleep to clear away the solutes that are present within brain tissue, and suggesting that there may be sort of an accumulating debt of clearance that can occur even with a single night of sleep deprivation. So this study was important in that it was the first human confirmation that there is a sleep-active clearance of solutes out of brain tissue. So it's happening both in humans and in rodents. So another biological piece of this story that's emerged over since actually our initial publication was the important role that astroglial water transport, and specifically the role that the water channel aquaporin 4 plays in this process of perivascular exchange or in glymphatic function. So if you're not familiar with aquaporin 4, or if you're not familiar with aquaporins generally, here's a quick primer. So aquaporins are a transmembrane channel that permits the rapid diffusion of water down its concentration gradient. Aquaporins are present frequently in secretory epithelial cells where they're involved in fluid secretion. They're expressed obviously in the kidney where they're involved in the regulation of fluid and solute dynamics. Within the CNS, we principally think of there being two aquaporins expressed. So aquaporin 1 is expressed in the choroid plexus, where it is involved in the secretion of cerebrospinal fluid. The other aquaporin that's present, that's dominantly present within the CNS, is aquaporin 4. So this is an immunofluorescence labeling from a rat, and where aquaporin 4 is labeled in purple, the astroglial marker GFAP is labeled in green. And what you might think from this image is that aquaporin 4 is expressed by vascular cells, perhaps by the vascular endothelium. But work, particularly initially, it was electron microscopy work, but more recently, bulk and single cell RNA-seq has demonstrated that aquaporin 4 is actually a highly astrocyte-specific gene product. So it's actually one of the more astrocyte-specific gene products that there is. So while it's expressed in the astrocytes, its localization is very peculiar. It localizes mainly to the part of the astrocyte that forms the barrier with the cerebral vasculature. So if I'm a capillary, if I'm this capillary, for example, looking out at the brain, somewhere between 70 and 95% of the surface area that I see is actually made up of perivascular astrocytic end feet. So the capillaries, they don't look out typically on neurons, they look out on these astrocytic end foot surfaces. And somewhere around 50% of the surface area of these astrocyte end feet is occupied by dense crystalline arrays of these water channels, which has been a little bit of a mystery in the glial cell biology field for a long time. So the brain vasculature is notable for its relative water impermeability to water. So it doesn't express other aquaporins. It doesn't have rapid pinocytosis. It's not fenestrated. So it seems to be specialized to control solute and water movement. And yet it's opposed by these perivascular astrocytic end feet, which seem to be specialized for rapid water conduction. And that's been a little bit of an enigma in the glial field for a long time. So our thinking is that what aquaporin 4 is doing is that it is helping to organize water movement through brain tissue, so diffusive water movement through brain tissue along the axis of the vasculature. So if you imagined the brain tissue without aquaporin 4, you would imagine that water diffusion. So if I took a look at this astrocyte and filled it with a bunch of water that was labeled, the water within this astrocyte would diffuse, the word is isotropically, meaning equally in every direction. It would go this way, and it would go this way, and it would go this way, and it would come out of the screen and into the screen equally. But in the presence of aquaporin 4, water diffusion through this astrocyte, while it can occur in any direction, it will be preferential toward these perivascular spaces, because there's an easier path for diffusion into the space through these aquaporin 4 filled end feet. So our thinking is that perivascular aquaporin 4 supports water and solute movement through brain tissue through this perivascular localization. So in the initial characterization of the glymphatic system, almost 10 years ago now, we tested the role of aquaporin 4 in this process. So we looked at animals, wild-type animals, we looked at aquaporin 4 knockout animals. So these are animals where the gene encoding aquaporin 4 has been disrupted. And we measured the movement of CSF tracer into brain tissue. So this is looking at the movement of a 45 kilodalton protein tracer into brain tissue after 30 minutes. And what we observe is in the aquaporin 4 null animals, the rate of tracer influx into brain tissue is markedly reduced compared to wild-type animals. We see the same sort of slowing if we measure the clearance of solutes out of brain tissue. So whether we're injecting the inert tracer mannitol or radiolabeled amyloid beta into brain tissue, we can measure its clearance out of the wild type brain. So for example, amyloid beta, about 70% of the injected amyloid beta is cleared in the first hour out of the wild type brain. But when we look at the clearance of either mannitol or amyloid beta out of the brains of wild type or aquaporin 4 knockout mice, we see that the rate of clearance is markedly slowed. So both the movement of CSF into brain tissue and the clearance of interstitial solutes out of brain tissue, it seems to be dependent in some way on aquaporin, on the aquaporin 4 gene product, so on this astroglial water channel. So if glymphatic function, if perivascular exchange is involved in the clearance of amyloid beta, and what I will tell you but not show you because of time, is that it has also been implicated in the clearance of tau, which is what neurofibrillary tangles is made up of. It's also been implicated in the clearance of alpha-synuclein, which is what Lewy bodies, which is the pathological feature in Parkinson's or Lewy body dementia. So all of those proteins which aggregate in the setting of neurodegenerative disease appear to be passengers in these perivascular spaces, and this perivascular exchange appears to contribute to their clearance. So if that's true, then is it possible that that an impairment of this process, an impairment of perivascular exchange, is part of what underlies the vulnerability of the aging brain or the injured brain to the process of protein misaggregation and the development and progression of diseases like Alzheimer's? So this is a fairly straightforward study where we simply tested the question, asked the question, what is the effect of aging on this process? So if we take animals with really nothing else wrong with them other than the fact that they're old, and we measure the effect of age on perivascular exchange, what do we see? And the results are actually fairly striking. So when we look at the movement of two different tracers in this case, so this is a 3 kilodalton tracer and another 45 kilodalton tracer, one's red, one's green. We look at their distribution after 30 minutes. We see that in healthy young animals, so 2 to 3 month old mice, we see pretty extensive and pretty rapid distribution of CSF tracers through brain tissue. And we see in aged animals, so in 18-month-old, which is late middle age, sort of early elderly mice, we see that that process is markedly reduced. When we measure the clearance of solutes out of brain tissue, we see the same sort of thing. We see a parallel process. So if we measure the clearance of amyloid beta 40 out of brain tissue in healthy young, so 2 to 3 month old animals, we see that in the first hour, about 70% of the amyloid beta has been cleared. When we look at 12-month-olds, so middle-aged animals, we see that that process is slowed. And when we look at 18-month-olds, so pretty old mice, we see that that process is slowed further still. So what that tells us is just in the presence of aging, so with no concomitant pathology, we see that the process of perivascular movement in and the clearance of solutes out of brain tissue appears to be impaired. So what is it then that's changing? What's changing? What are the changes that underlies those changes? And just sort of by way of a little bit of orientation here, and I'm obviously not going to go into this, but similar impairment in the clearance of glymphatic function has been observed in rodent models of hypertension, diabetes, traumatic brain injury, ischemic injury, and in transgenic mouse models of Alzheimer's and Parkinson's. Importantly, all of these features are either models of or models of risk factors for Alzheimer's disease and other neurodegenerative diseases. So it appears that that impairment of this process is common to many of the different risk factors for neurodegenerative diseases, including Alzheimer's. In the limited number of studies that have evaluated glymphatic function in human subjects, so there's only been a few studies to date, there's evidence for impairment of glymphatic function in normal pressure hydrocephalus, in cerebral small vessel disease, and in idiopathic intracranial hypertension. So is it possible, so if aquaporin 4 supports glymphatic function, and if glymphatic function is impaired in the aging and in the injured brain, is it possible that changes in aquaporin 4 expression are what's going wrong? So our initial hypothesis was actually that perhaps aquaporin 4 expression was declining with age, and that would explain the reason why we see impaired glymphatic function. But that's not what we saw at all. So this is results from a Western blot quantification of aquaporin 4 expression. So either total aquaporin 4 expression or the expression of the two characterized aquaporin 4 isoforms, the M1 and the M23 isoforms, in 2 to 3 month old brains or in 18-month-old brains. And what we observed is that aquaporin 4 expression doesn't decline with age, rather it increases. But what we see is that while its expression increases, its localization changes. So while in the healthy young cortex, we see that almost all of the aquaporin 4 local expression is localized mainly to these perivascular end feet. So at the level of a single astrocyte, you can see that almost all of the aquaporin 4 localizes to the processes that surround the cerebral microvasculature. When we look in the aged animals, we see more global immunofluorescence, but we see that the localization has sort of expanded. It's no longer restricted solely to the perivascular end foot elements, but rather we see that the aquaporin has begun to distribute to all of the fine processes across the astrocytes. So we imagine that either the loss of perivascular localization or maybe this gain of non-perivascular localization is changing the ability of the astrocytic network to organize fluid movement along the vasculature. So we began to look experimentally at these two different processes, the loss of perivascular localization and the gain of non-perivascular localization separately, to try to get a sense of whether one or the other was involved in the impairment of glymphatic function that we observe in the aging and the injured brain. So in the first series of experiments, what we begin to look at is whether this gain of non-perivascular localization is the problem. So to do this, we use an AAV, so an adeno-associated virus-based approach. So we use the PHPB AAV variant to drive aquaporin 4, a specific isoform that tends to be non-perivascular, under the control of the of modified human GFAP promoter. We drive expression across the brain of animals. And so using this approach, we can get pretty global transduction of astrocytes throughout the cortex and other brain regions. And the interesting thing is this, you can sort of see the splotchy localization of aquaporin 4 is a pretty decent phenocopy for this kind of non-perivascular localization of aquaporin 4. So when we drive non-perivascular localization of aquaporin 4 and then measure glymphatic function, what we observed is that there's no real effect on this perivascular exchange. And when we measure the changes in these kinds of changes in aquaporin 4 localization on the development of amyloid pathology in a transgenic mouse model that spontaneously forms amyloid beta, we see no effects on the development of amyloid pathology, whether we measure amyloid beta 40 or 42, whether soluble or insoluble. So these data tell us that this gain of non-perivascular aquaporin 4 doesn't appear to affect lymphatic function, and it doesn't appear to affect the formation of amyloid pathology. In a second experimental approach, we evaluated whether it's the loss of perivascular localization, that's the other feature that we observe, is contributing to the impairment of glymphatic function and the development of Alzheimer's-related pathology. So we know that aquaporin 4 maintains its perivascular localization through its association with the dystrophin-associated complex. So that's a macromolecular multi-protein complex. It includes alpha-syntrophin, dystrobrevin, dystrophin, and dystroglycan, which anchor it to the basal lamina. So it anchors it to the matrix at the cerebral circulation. If you delete any of these genes, so whether alpha-syntrophin, dystrobrevin, or dystrophin, what you get is this phenotype, and the image isn't really coming through well here, but essentially you get normal expression levels of aquaporin 4, but it loses its perivascular localization, right? So it ends up, it doesn't cluster around the perivascular end feet. And what we observe is that in the absence of perivascular localization of aquaporin 4, we see an impairment of glymphatic function. And when we cross the alpha-syntrophin null line into the background of this transgenic Alzheimer's disease model line, so these are the TG2576, this is the TG2576 line, and we measure amyloid beta 40 and 42 levels in animals with, that's in red, or without alpha-syntrophin, so those with perivascular localization of aquaporin 4 and those without, we see that there's both an impairment of glymphatic function and an exacerbation of amyloid pathology that we observe when aquaporin 4, perivascular aquaporin 4 localization is lost. So what these data tell us is that the loss of perivascular aquaporin 4 localization, and not this gain of non-perivascular localization, appears to be capable of impairing glymphatic function and driving the development of Alzheimer's-related pathology. in data I'm not going to show you, this alpha-syntrophin gene deletion also appears to exacerbate tau pathology and alpha-synuclein aggregate propagation. So we see that this effect isn't just on amyloid, but it's also on the development of tau and synuclein pathology. Don't have time to show you that data. So everything to this point talks about the role of aquaporin 4 in the impairment of glymphatic function and the development of Alzheimer's related pathology in rodent models. But that doesn't necessarily speak to whether it's involved in the pathogenesis of human Alzheimer's disease. And so the last little bits I'm going to tell you, and I'm going to sort of zip through here so we can get to questions, is how do we evaluate that in humans? So obviously, doing this experimentally is tricky because human clinical populations, it's much more difficult to get at causal relationships. But what we've been able to do is look in different human data sets for corollary data that at least supports a possible role of aquaporin 4 in glymphatic dysfunction in the development of human Alzheimer's disease, even though we can't do the types of experiments that we do in rodents where we can actually modulate aquaporin 4 expression or localization to drive disease. So this is the results of a histopathological case series that we conducted. This is in collaboration with the Oregon Alzheimer's Disease Center Brain Bank. So this is a case series principally of elderly subjects. These are people over the age of 65, and they represent a spectrum of Alzheimer's related neuropathology. So from people who are cognitively normal to people who have histopathologically confirmed Alzheimer's disease, and including people with a clinical diagnosis of mild cognitive impairment, which is sort of this intermediate waypoint that where people sort of sit for a period of time, frequently progressing to Alzheimer's disease later. And what we observe is that if we measure perivascular polarization, so the amount of perivascular aquaporin 4 to non-perivascular aquaporin 4, what we observe is that there's a loss of polarization among subjects with histopathologically confirmed Alzheimer's disease, and we see that the subjects with MCI exhibit an intermediate phenotype. So perivascular polarization appears to be, or the loss of perivascular polarization appears to be present in the setting of human Alzheimer's disease. We can evaluate whether these changes in perivascular localization correlate with either cognitive and functional decline or the presence of Alzheimer's related pathology. The interesting thing that we observe is that the cognitive the associations that we observe are only in the earlier phases of the disease. So when we look at subjects who are either cognitively intact or have a clinical diagnosis of mild cognitive impairment, we see that indeed loss of perivascular localization, which is heading to the left here, is associated with a lower score on the mini mental state examination or with a higher score on the clinical dementia rating sum of boxes. So these are these are measures of global cognitive function and sort of functional independence. So we see that loss of perivascular localization is associated with cognitive impairment and functional decline. At least in the earlier phases of the disease, that association goes away when we look at end-stage disease. Similarly, when we look at local measures of local Alzheimer's pathology, whether it's the seroneuritic plaque score, so this is a measure of amyloid pathology, or phosphotau levels in the frontal cortex, we see that indeed loss of perivascular localization is associated with increasing amyloid plaque burden and increasing local tau burden in the earlier stages of the disease. And as the subjects approach end-stage disease, we see that association goes away. So what this suggests is that this loss of perivascular localization is occurring in AD. We see it there, but it associates most strongly with Alzheimer's pathology and cognitive decline in the earlier phases of the disease. I'm going to skip over this bit just to get to my final piece, which is that pathological association between Alzheimer's and Alzheimer's pathology and changes in aquaporin 4, we don't know which way the causation goes, right? So the pathology, the Alzheimer's pathology could be driving the changes in the astroglial compartment to drive changes in aquaporin 4, or vice versa. Changes in aquaporin 4 could be promoting Alzheimer's pathology. We can't get at that with a neuropathological study. So we've done a gene association study where we look at naturally occurring variants in human aquaporin 4, alpha-syntrophin, and dystrobrevin and dystroglycan genes. So these are the genes that are either aquaporin 4 themselves or who determine aquaporin 4's localization. So we evaluate whether tagged SNPs in those human genes, so we looked at 11 tagged SNPs in aquaporin 4, 40 in dystrobrevin, 4 in dystroglycan, and there's actually only one SNP in the alpha-syntrophin gene in the gene arrays we looked at, whether those SNPs are associated with endophenotypic markers of Alzheimer's disease pathology or clinical progression. What we observed, and I'm going to really condense down, so I'm not even going to go through the data with you in the interest of time, but what we observe is that SNPs, both in the aquaporin 4 gene, the dystrobrevin, and the alpha-syntrophin gene, SNPs across these genes are significantly associated with CSF biomarkers of Alzheimer's disease, so either amyloid beta 42 or phospho-tau levels. We see that SNPs in the aquaporin 4 and dystrobrevin gene are associated with longitudinal measures of cortical atrophy measured by MRI. And these are all data taken from the Alzheimer's Disease Neuroimaging Initiative. We see that SNPs in the aquaporin 4 dystroglycan and dystrobrevin gene are associated with measures of cortical atrophy, or I'm sorry, hippocampal atrophy. And we see that SNPs in the dystrobrevin gene are associated with longitudinal cognitive decline. So what these data suggest is that variation in the genes that either encode aquaporin 4 or encode the elements that define its localization, they seem to be conferring either risk to or protection from the development of Alzheimer's pathology or the corollary markers of its clinical progression, whether that be atrophy or cognitive decline. And that is at least consistent with the role of this biology in the development of human disease. So by way of summary, the glymphatic system, if this is new to you, this is the cartoon version. Obviously, the cartoon version is a cartoon version and not the real version that we have in our minds, but it's a good place to start. It is a brain-wide perivascular network that supports waste clearance through the exchange between the CSF compartment and the brain interstitial compartment. It appears to be primarily sleep active, and it appears to be dependent on the perivascular astroglial water transport. Some things we know. We know that glymphatic function appears to be impaired in the aging and the injured brain, both in animal models and in emerging data from human subjects. We see evidence for changes in aquaporin 4 being associated with the development of human neurodegenerative disease that supports a role for the dysfunction of this biology in human disease pathogenesis. We see that on human postmortem histological studies. We see that in human transcriptomic studies, and now we see it in human SNP association studies. Based on these findings, where we are at right now, we believe, and I think others are beginning to as well, that impairment of glymphatic function appears to be a general feature of the aging and the injured brain. It seems to be a common pathway, and that loss of this clearance, including in the clearance of amyloid beta, tau, and synuclein, appears to be a host feature that makes the brain vulnerable to this the process of protein misaggregation, and that this may be contributing to the development actually of a wide range of neurodegenerative diseases. So from Alzheimer's disease to Parkinson's to Lewy body dementia to chronic traumatic encephalopathy. And so by either targeting this pathway in a prevention setting or in a treatment setting may be viable. And alternatively, the biomarkers that indicate this biology, because this biology sits upstream of the development of amyloid, tau, or synuclein pathology formation, may actually give us a sense of to what extent a brain is vulnerable to these processes. So it may be a way to define vulnerability to the development of these sorts of settings. So with that, I'd like to give thanks to my present lab members at the University of Washington, two former lab members, Matthew Simon and Marie Wong, who are no longer with our lab. I showed a whole bunch of their data. They've moved on to bigger and better things, but I definitely want to acknowledge them. And I want to acknowledge lab funding from Department of Defense, Veterans Affairs, and of course the NIH. This is my lab's website. This is my email. If you want to contact me, I'm glad to chat. So I will go ahead and stop talking and answer questions now. So thank you.
Thank you so much. That was wonderful. So yes, audience, if you have any questions, please use Ask a Question tab and you can post now and I'll ask some. But yeah, you can unshare your screen if you'd like.
We have infinity on the screen again. Okay.
There you go. So I'll start with a question that I had. I thought it was interesting when you noted that various types of injuries are involved or result in glymphatic dysfunction, and one of them was including cortical spreading depolarization. For that, I think of like I don't know that much about it, but I think of sort of like seizure altered activity, right? So do you know whether altered like neuronal activity in other cases
Yeah.
also
So
alter glymphatic
I think
function?
that's probably the most interesting question nascent in the field right now. And it's a question that is people are just starting to think about and just starting to experimentally address. So cortical spreading depolarization is sort of it's a strange phenomenon, right? And one of the key features of it is that there's this really striking compression of the extracellular space that results from cellular swelling. And so one might imagine that the effect of CSD specifically on interstitial solute movement results from the changes in extracellular volume fraction. That's a big change, and the vasomotor changes that come along with CSD, right? So I view that as a little bit of a fringe case, but pathologically important in the setting of Alzheimer's disease, or not, I'm sorry, migraine. Um, I think the more
Oh,
interesting question, if you think about, so CSD is, we think of it as a fringe case. So you essentially have this closing off the extracellular space that follows the progression of the CSD wavefront, right? So that's neural physiomechanical like compression, right? That's being driven by neural activity. Well, suppose we dial back that the impetus from a CSD wave to what about a slow wave? Right? So slow wave. So the period of sleep that's has been most implicated in glymphatic exchange is slow wave sleep. So N3 sleep, which is characterized by low frequency, so typically less than 1 hertz, synchronous oscillations between upstates and downstates. So upstates of synchronous activity and downstates of synchronous inactivity. So if you think about, as a slow, like when an upstate is present in a local area of tissue, right? So think of microscopic tissue domain. Well, that synchronized activity is going to be associated with synchronized potassium uptake and synchronized cell swelling, and probably synchronized compression of the extracellular space. Not CSD level compression, but compression, right? And then that upstate is then followed by a downstate. Well, that downstate is associated with synchronized cell shrinkage and synchronized expansion of the extracellular space, right? So one might imagine that while CSD is this, really intense, like tractoring, movement of tissue or movement of fluid and solutes to the extracellular space, well, slow wave activity may cause the same sort of thing, right? So it may allow the
yeah.
coupling between synchronized neural activity connecting to fluid solute movements. There were data from Laura Lewis that came out about 2 years ago now, suggesting a link between K complexes, which are isolated slow waves, right? Hemodynamic variation that were phase locked or that were time locked. They weren't necessarily in phase or just sort of phase lagged. And then fluid dynamics in the ventricular system argue that such neural, vasomotor, physiomechanical coupling is at least possible macroscopically. So they were looking at CSF dynamics in the ventricles. Now it's possible that the same sort of process is actually happening writ small throughout the neuropil during different electrophysiological events. So that was it, like a
Yeah.
10-minute answer to a 30-second question.
I mean, it's super fascinating when you think of, I don't know, seizure as well. Like, how does that affect the vasculature and in
Absolutely.
that case? But very
Absolutely.
interesting. Thank you. So I have a few questions here. So someone asks, Rita asks, is it known if changes in sleep patterns affect aquaporin expression and localization?
Yeah, so we're just starting to think a little bit about this. So there's one piece of data out there. We can think about that across acute, moment by moment states, and we can think about this chronically, right? And so there isn't a lot of data in either space, but it could be occurring either way. So, as an example, when you talk about sleep disruption, well, people with obstructive sleep apnea, right? People who are old who have poor sleep. Those people may have alterations in aquaporin 4 that result from the neuroinflammation that occurs as a result of chronic sleep disruption or chronic OSA or obstructive sleep apnea, right? So you can think of sleep disruption occurring chronically as a neuroinflammatory stimulus that will have a range of effects, among which are changes in aquaporin 4, right? And other sort of fluid and solute handling. So that's sort of one way to think about it. And so we're actively pursuing that right now. So we have a case series of people where we have 115 subjects who have had sleep characterized and who have come to autopsy, right? And so we can start to look at the relationship histopathologically between pre-morbid sleep behavior and post-mortem neuropathological outcomes. That's one way to look at it. I think the more interesting question is whether between different sleep stages or different maybe elements of the circadian cycle, there's a similar type of regulation, but that's occurring rapidly. And so the only data at this point that's been published is from Lauren Hablitz's paper that described the circadian regulation of glymphatic function. So this was Hablitz, I think it was Nature Communication. I'd have to check on that, but it was from Mike Niedergaard's lab. It was about 2 years ago, a year and a half ago. But what they observed, it's like buried in one of the figures, but what they do see is circadian regulation of these perivascular infant elements. That it seems to be, you see changes both in transcriptional regulation and in localization. So it may be that there's much more rapid and dynamic regulation of that localization than we previously thought.
I see. So this kind of links to another question in the chat, which maybe you sort of answered it, but they asked if there's a difference in the efficiency of clearance in the animal in different phases of sleep, so NREM and REM.
Yeah. So
that might be difficult to study.
It's well, actually, so this is one of the, I think we're moving in our field. So right now, so when I came to the University of Washington 3 years ago, our lab was 80-20 rodents, humans. In terms of the volume of work we're doing. Right now, I would say we're 40-60 rodents, humans. And I think over the next 5 years, we're going to move probably toward 20-80 rodents, humans. I think we're at a phase in this biology where, partly because of some of the scale issues, right? So when you're studying diffusion, distance matters a lot. And so evaluating things that are occurring things that occur in the time scale of seconds in a mouse occur in a time scale of hours in a human. And so some things are actually easier to capture in a big human brain. Looking at differences between sleep-wake states in rodents is really tricky because rodents have really weird sleep. they regulate sleep really strangely, right? And so they have very fragmented. They don't sit in these really consolidated sleep states. They're in and out of all of these different their different sleep stages, and so it's kind of tricky. Whereas in human studies, you can sleep deprive a person and bring them in and put them in the MRI magnet in the morning, and they'll drop into N3 sleep right away in recovery sleep, right? And so we actually can, we can get a fair amount of consolidated N3 sleep. Now, the downside to that is actually getting REM sleep can be a little bit tricky because recovery sleep is very N3 dominant and very sort of REM light, right? And so right now the data suggests that there's a strong component of delta activity is a key driver. Whether it's more active or not during REM, I think the jury's still out. There's an experimental issue that REM is a little bit hard to capture in humans, like in a magnet, which is typically where this is being evaluated.
Got it. Great. Well, it's 9:01, so we can stop there. I don't see more questions, but if anyone would like to join us for this Zoom talk, we're going to take about a 5-minute break. We'll say, please join, by like 9:07, 9:10 in the Zoom room if you'd like to meet with us and the speaker. And so thanks again for that really great talk. And we look forward to speaking further. And for everyone, if just a heads up, we don't have a seminar next week, but our next one will be in 2 weeks on November 8th when we host Dr. Kevin Tracy from the Feinstein Institutes for Medical Research. So please check out our webpage, and thanks for attending, and thanks for the talk, Dr. Iliff.
Thank you very much.
I'll see you soon. Thanks.